All right, good afternoon, everyone. Let's keep going with our class. So you get a quiz back, and hopefully you're able to review it and see, where you got wrong. And as I said, your next quiz is going to be next Tuesday, and it will cover the second half of lecture 2, which includes proteins and nucleic acid, as well as all of number 3. I'm going to go through a majority of number 3 today, and whatever we don't finish today, we will do on Thursday's class. Okay. So quiz 2, I already posted a review video for you. You can once again log on to Luminate and go to the courses. You guys are 202. So over here, right? Quiz 2, you will see the quiz 2 information. There is a review video for it. And I will enable this at a later time for you. How many questions are we talking about? We are going to have 15 multiple choice questions, 4 short answer questions, and 5 labelings. We're going to do the labelings maybe today, maybe next class. I don't know. But it's going to be this one. All right. It's from the study guide. So you will either get an animal cell or you will get a plant cell, and it's going to be the exact same one. Okay. But without the words. Okay. You have to know how to spell them and stuff like that. Okay. So start practicing on those. If you didn't do so well on the first quiz, like I said, there are multiple resources available to you. The study guides are very useful. The review videos are very useful. Tutoring service is also available to you free of charge, right? Just have to book it with the library. You can also go to the library and ask to see a learning strategist. They will give you various tools to help you prepare for quizzes. And of course, if you have content-specific questions, you are more than welcome to email me. One thing I want to emphasize is if you need to find me, do not use the message function here, okay? If you use the message function, it takes too long for me to get your message, okay? Because I don't check this all the time, but I have my phone with me pretty much all the time. If you email me, I will get it right away and I'll reply you. Okay, those of you who have emailed me know I get back to you very, very quickly. My email is at the beginning of every slide deck. And if you must know, there is contact info for me right here. Okay, it's vwong@centennialcollege.ca. All right, let's get to it. So last class, we went through the cell theory. There are 4 parts to it. All living things are made up of 1 or more cells. We talked about how the cells are individually alive out of the organism. And we talked about all cells come from the division of pre-existing cells. And lastly, we talked about how genetic materials are passed on to the future generation as a cell divides. So the focus for today is to learn the various structures that you will find within the cell. And let's start with the different categories of cells. So when it comes to cells, we generally separate them into 2 main categories. We have what's called eukaryotic cells, which are also called eukaryotes. And these are cells that are typically bigger. They have a special circular structure in the middle called the nucleus. And that's where you will have the genetic material. They have many different types of organelles, right? Individual structures within the cell that perform specialized functions. And animal cells and plant cells are examples of eukaryotes. Okay, so you are made up of eukaryotic cells. Almost all the cells in your body will have a nucleus. Now, I just want to briefly mention to you that animals and plants are not the only thing that exist on this planet. We also have those things called protists, right? Okay, so an example of a protist would be an amoeba, and there's something called paramecium. Yeah, so those things are part of a separate category. You will see them come up a few times here and there. Okay, so the protests are also examples of eukaryotic cells. Generally speaking, they're just more complicated, they're bigger, and they have the most distinctive feature that separates them from the other category is the presence of a nucleus. So the other type of cell would be prokaryotes. Okay, prokaryotes. Prokaryotes are typically smaller and simpler in size and in structure. They do not have a nucleus to keep their genetic material. Okay, it's there, but it's not protected within a specialized compartment. They don't have a lot of organelles, just a few essential ones to keep them functioning. And example of prokaryotes would be bacteria. You guys know names of any bacteria that you might have read on the news, you know, when they recall salads and whatnot? What are some names of bacteria? Salmonella, yeah, good Salmonella. Anything else? Ebola is a virus, but a good try. What's that? Lactobacillus, yeah. That one is a good one though. Okay. It's in yogurt. Okay, you guys are being very fancy. Okay, I'm just looking for very simple ones, guys. Right? Recall of like, you know, salad or even sometimes water sauce get contaminated. This it starts with one letter followed by a short name. No? E something? E. coli. There you go, right? You guys heard of E. coli, right? No? Yeah? Okay. If not, watch more news, guys. Okay. E. coli, just kidding. E. coli is it's a bacteria that can cause severe diarrhea, for example. And sometimes they do make themselves into our food source. But the point is, these are very primitive, simple cells compared to eukaryotic cells, and we call it prokaryotic cells, okay? Or prokaryotes. Okay. So what are we? Are we pro or eukaryotes? Eukaryotes, very good. That's what we are made up of. Okay, so all cells are surrounded by the cell membrane or the plasma membrane. And by now you should know the main component of the cell membrane are these things called phospholipids, right? Phospholipids are special because they have a hydrophilic head and a hydrophobic tail, right? And they form two layers, kind of like a sandwich, to make up the cell membrane. Okay, and you can see it right here. The cell membrane separates the surrounding of the cells from its content, and it has properties that allows it to become selectively permeable. Selectively permeable means it only allow certain materials to cross. You don't want everything to be able to pass through the cell membrane. You want to keep like harmful things out of the cell, and you want to keep nutrients inside the cell. So there must be some kind of regulation and some kind of control as to what can go into the cell and what can leave the cell. And the cell membrane has the ability to help regulate. Okay? It's kind of like the security person at the front desk of a building. It checks to see who can come in and who cannot. So this is another view of the phospholipid bilayer, and you can see the tails are all sandwiched in between. We actually will draw this out in much more detail in lecture 4. If you look at an actual picture of the cell membrane, it's right here. That's the cell membrane. This would be the outside of the cell, and this would be the inside, which we already know it's called the cytoplasm. And these two lines here are basically the phospholipid bilayer. Okay, the darker black line is the hydrophilic head facing either outside or inside, and the gap in between is where you will find the hydrophobic head. Okay, so much of it is actually hydrophobic, with a little bit of hydrophilic on either side. Think of it as peanut butter sandwich. Okay, you have the bread on the outside, both sides, and then you have the peanut butter in the middle, forming the hydrophobic layer. So what we're gonna do is we're gonna be drawing quite a bit today. Based on what people tell me in the past, drawing it out helps them understand it a little bit better. So if you can please take out a piece of paper, that would be wonderful. If you have colors, even better. If you don't have colors, please consider getting some colors for this course. This is not going to be the last time we're going to be drawing things, okay? I'm going to be drawing a lot of things for the whole course. And colors would be preferred. If you have one of those tablets, of course, you will not run out of colors. All right, I'm going to give you an idea of what we are drawing so you can plan accordingly. I drew this with my other class. This is what we're doing, okay? Yeah, it's very, very lovely. Yeah, I think. So take out a blank piece of paper, turn it sideways, and here we go. Okay, so we're going to be drawing a eukaryotic cell. I just want to remind you, eukaryotic cells are bigger, more complex, and the defining feature that separates it from other cell is the presence of this thing in the middle. What is the name of that? The nucleus, that's right. So all of our cells, with the exception of a few of them, will have a nucleus. Okay, so what we're going to do, we're going to zoom in right here with our arrow, and I'm going to be drawing a nucleus. Okay, so think of this as a 3D ball, all right, 3D shape, and I'm going to cut it open a little bit. Okay, I'm going to cut this thing open, but not all the way. I'm just going to cut like a wedge of it. Can you visualize this as a 3D structure? There we go. Okay, it doesn't have to be beautiful. It just has to be similar to what you see. And I encourage you to reorganize it when you go home anyways. Okay, so this right here, this is the nucleus. Nucleus. Now, if you have a highlighter, which would be great, you can kind of shade out the nucleus a little bit here, okay? So the nucleus is actually covered by a membrane. Okay, so I'm drawing the membrane that wraps around the nucleus. Now, because I cut it open, right? So this portion is going to be missing the membrane. Okay, so this here, this is called the nuclear envelope. The nuclear envelope. And what it does is it surrounds the nucleus. Walmart has crayons on sale, okay, $1.99. I just bought a pack for my kids, okay? So you can go and get some if you need to. Now, we don't want to seal the whole thing, okay? We don't want to seal the whole thing. We want to have some openings for things to enter and leave the nucleus. And so there will be some pores, okay? These are little holes that allow things to enter and leave the cell. Okay? These are called nuclear pores. nuclear pore. And basically they allows materials to enter or leave the nucleus. And you might be wondering, what kind of things are gonna enter and leave the nucleus? We're gonna get back to that in a second. Is it too small, Ruben? You cannot see? Yeah. Here, I'll try to make it a little bit bigger. There you go. So on the inside of the nucleus, there is an other round structure. Okay, like it's the whole thing, but we are only showing a little bit of this guy. And this thing, let's color it brown. It doesn't really matter what the colors are. This is called the nucleolus. Now, once again, the nucleolus is inside the nucleus. Okay, and the function is actually to produces another organelle called ribosome. Okay, we'll come back to the ribosome in a second. So once they make the ribosomes can leave the nucleus through the pores, right? Through the openings allow things to come and go, right? In the nucleus. Okay, what about this region here? What would you find inside the nucleus besides the nucleolus? You will find genetic material, okay, in the form of chromatin. Okay, chromatin is basically DNA wrapped around proteins. Okay, DNA wrapped around proteins. Chromatin, chromatin, chromatin. So if you look at the DNA inside one cell, how long do you think your DNA is going to be for one cell? If you just lay out the DNA fabric, give me some wild guess. How many? Did you say centimeter?
Same number.
Oh yeah, same number, but like how long would it be? How long would it be? Okay, nobody knows, that's okay. It's 2 meters. Okay, 2 meters of DNA per cell. Okay, 2 meter is like, I'm not that tall, but then 2 meter, I guess it's up here, right? From the floor up to here. Right? And you gotta pack all those DNA into a tiny nucleus. And the nucleus is not too big, right? Remember, we measure these things in micrometers, right? Millions of meters. So how do you pack them together? Okay, so I'm gonna explain that to you. And people always get confused with this thing, so I'll try. And first of all, you already know DNA, right? I hope you learned it from me last week. So that's the double helix, and we have the base pair. And this is DNA by themselves, just on its own. We call it naked DNA because it's just DNA with nothing. Okay, naked DNA. Like I said, you get 2 meters of this stuff, very long. It won't fit into the nucleus if you don't do something about it. Right? So yeah, well, it's already zipped, right? Okay. Because it has two strands, right? But is there anything else you can do in addition to zipping it? Well, you're gonna take these DNA and then you're gonna wound them up around some special packaging proteins. Okay, so there is the packaging protein right here. These little um, orange circles, these are proteins. Okay, proteins. It's like a yo-yo. Think of the yo-yo. The string is the DNA, and the circle thing is like the protein. Right? The string is really long, right? But if you wrap it around the yo-yo, then it's very compact. And so imagine taking a DNA and wrapping around multiple proteins, multiple yo-yos, then you can store the DNA nicely in a compact format, right? So that configuration is basically what the chromatins are. And chromatins. So chromatin is this stuff here. Now, most people have not heard of chromatin before, unless you take biology. But I'm sure you would have heard of chromosome, right? Chromosome. So what's the difference between chromatin and chromosome? Well, actually, if you take the chromatin, you can further condense it. Okay, you can condense it even more, right? Very, very packed, very, very compact and condensed. Okay. And you do it, over and over and over again. And then a lot of times when it's already in this form, it's going to have 2 of them. Okay. It looks like I'm just like scribbling, but hold on, it will make sense. And then you will get this thing in the end. And that's what people are used to seeing on movies and whatnot, right? The X, right? This is the chromosome. So chromosome is essentially same as chromatin. It's DNA with proteins, but it's more compact. Okay, like you take this stuff and then you crunch it more, take out more space, and then you make it even more condensed, then you get chromosome. Okay? So when the cell is not dividing, then the genetic material is going to exist in a chromatin format. But if you need to split the cell, if you need to copy the cell, then you need to move the genetic material around, right? Then you gotta pack everything into chromosome form before you move. It's like, moving from one apartment to another. Before you move, you gotta pack everything, right? And then after the move, you can, take everything out again. And that's sort of what's happening. And you gotta pack the chromatin into chromosome before you move them. Once the cell finishes dividing, you can unpack them back into chromatin. So that's pretty much the nucleus and everything. Okay. Now I'm gonna give you an analogy to help you understand this. You don't have to draw it. Okay, just in case you're still a little bit confused. Okay? I like to relate things to food. It makes more sense for most people. So you can think of an avocado, okay? You don't have to draw this. Okay? Don't draw this, just watch. So this is an avocado. And then we have the seed right here in the middle. And then we're gonna have the meats, right? There we go. So if the whole thing is the nucleus, what would the seed be? The nucleolus, that's right. All right, the nucleolus. And what will we find in the meat right here? What are we gonna find in the meat? That's right. That's where you will find the chromatins, okay? Chromatin. And then the shell that you don't eat, I hope you don't eat that. That is the nuclear envelope, right? And then now take a fork or a toothpick, and then you poke a bunch of holes, and you get the nuclear pores, right? You understand? That's the nucleus and everything associated with it. Yeah, you don't need that. Oh, you want that? Sure. Oh, yeah, but this is not important. Here, I mean, it could be important, but I'll put it on the side, okay? I'll put the avocado to the side. It will be there when I post the notes. There we go. There we go. Okay, so we don't need that for now. We're gonna come back over here. Okay, so let's go through the PowerPoint slide briefly. It's basically what I've told you, but here are all the slides for it, right? Okay, so once again, nucleus, only eukaryotic cells have it, bacteria do not. And the nucleus is surrounded by the nuclear envelope. And actually, the nuclear envelope is going to extend outward into the cytoplasm and form something else. We're going to get to that later. On the envelope, you will find the pores. With professional drawing right here. And things can leave and enter through the pores. Inside the nucleus, once again, you will find genetic material, but not in its naked form. Right? The DNA will wrap themselves around happy protein. They will condense further to form chromatin. And then later on, if you need to move them, they will form chromatin. Okay, it's just the same thing, but different layer, different levels of compactness. Inside the nucleus, once again, you have the nucleolus, which is responsible for making ribosomes. Okay, so we have not drawn the ribosomes yet, but in this picture, you can see the yellow dots are ribosomes. So you will make it in the nucleolus, they will come out, and they will, some of them will stick to this other structure. Okay, so let's take a look at the ribosomes a little bit more. Even though the ribosomes look really small in this picture, if you zoom in, the ribosome is actually made up of two subunits, the large and the small subunit. Think of it as a burger bun, okay? The top bun and the bottom bun. Top part is bigger, lower part is smaller. And the ribosome itself is made up of two things. It's made up of proteins and it's made up of RNA. Okay, so RNA is like DNA, but it has the uracil instead, right? And the function of the ribosome is to produce proteins. Remember, proteins are made up of amino acids, right? So the ribosomes are responsible for connecting the correct amino acids together, right? And then it will build a protein. So this is a real picture of what it looks like. All these fuzzy black lines, black dots rather, those are the ribosomes. I will never give you an actual picture to label. I will just give you, cartoon ones. It's much easier that way. Some ribosomes are going to be freely floating in the cytoplasm, like these ones. Others will be attached to a network called the endoplasmic reticulum. We'll put this on our picture later on. Okay, so they can either be on their own or they could be stuck to the endoplasmic reticulum. Now, before we go back to our drawing, I want to explain the central dogma of biology. And that sounds like a weird word, but basically it just means theme, okay? Central theme, central dogma of biology. Now, DNA is often called the blueprint of life, and it is because it contains the instruction to make proteins. It contains the instruction of how the cell would function. And that is why it's important to, pass on all the DNA when a cell replicates, when a cell divides, okay? But when a cell is just performing its normal function, when it's not dividing, how do we get the information from the DNA and use it to make the proteins that we need? Okay, so it turns out DNA is very big and you cannot take it out of the nucleus easily. It's not even going to fit through the pores, right? So as such, we will keep the DNA inside the nucleus. Think of the nucleus as the library, and the DNA is like an encyclopedia. Okay, so in the encyclopedia, there are a few pages that you really want to take home and use it for your studies or whatever. Right? You cannot take the encyclopedia out of the library. So what are you gonna do? Yeah, nowadays you're probably gonna use your phone, right? Okay, but before that, you would photocopy it, right? You would only photocopy the pages that you need, not the whole thing, right? So similarly, when I need the instruction to make a particular protein, you will make a copy of the section of the DNA that you need. Not gonna copy everything, just the part that contains the instruction to make whatever protein you want to make. So that is going from DNA. And then you're going to make a copy in the form of messenger RNA. This process of copying it is called transcription. A transcription. To transcribe something, it literally means to copy something, right? So both DNA and RNA are the same language. They are both nucleotides, and that's why you are transcribing. So the messenger RNA, the copy that you've made, will now exit through the pores. It will go into the cytoplasm. And then the ribosome, the burger, the top and the bottom subunit, will read the instruction on the messenger RNA and use it to build proteins. Okay? So from mRNA, you will decode the information and build protein, and this is called translation. It's called translation because it's a different language, right? RNA is nucleotides. What about proteins? What language is protein? What is it made up of? Amino acid, exactly, right? So it's a different language, different system. That's why it's called translation. And so every protein that you make in your body has to go through this process. It starts with the DNA. You're going to make a copy of it in the form of mRNA. And then you're going to build the protein off from that. Okay. So let's add those things to our drawing, but maybe do a poll first. All right, let's see. Good. Try again. Good, good. Not quite. Good. Yeah. We just put up the finger. Yeah, that's good. Good, good, good. Okay, good. Not quite. Anyone else? Okay, we're looking for prokaryotes. Prokaryotes have no nucleus, right? Okay. So liver cells. Liver cells are animal, right? So they have a nucleus. That's not it. HeLa cells, you guys remember the story I told you last class at the end? Yeah. It's from the cervical cancer from the lady, right? That is eukaryote. E. coli is a bacteria. That's prokaryote. Okay. Amoeba is a protist. That's eukaryote. How about this one? You've been paying attention, you should know this. What's the nucleolus do? Good. Yes. Yeah. Not quite here. Yes. Yes, let's see. Good, good. Anyone else? Not quite. Yes. Nucleolus. The nucleolus is the thing inside the nucleus. The job is to produce ribosomes. Okay? Produce ribosome. We wrote it down right here. See? Nucleolus produces another organelle called ribosome. All right, next, before we draw, we're going to be drawing a bunch of stuff that basically describes the endomembrane system. Endo means within. So this is a system that's made up of a bunch of membranes that's within the cell. The cell is surrounded by the membrane, but now you have like more membranes inside the cell. Collectively, they work together to produce and process and digest large molecules, okay? And they will also do a little bit of recycling and breaking things down as well. These are the key components of the endomembrane system. We already did nuclear envelope. Now we're going to do these things. Okay, so here we go. So, yep, same thing. Okay, so chatty, you guys at the back. Focus, please. So here, we're going to have the nuclear envelope, which is going to extend outward into the cytoplasm. Cytoplasm is all out here, right? So you can just randomly draw some tunnels coming out like this, okay? There we go. Okay, it's like a network of membrane. So this network is called endoplasmic reticulum. Now, I know I said I'm not too tight on the spelling, but some of the spelling were pretty off on the quiz, okay? So do try to improve over the semester, okay? Because next semester, I mean, you might be in my class again, who knows? But let's just say there are some teachers who are more picky about spelling, okay? So you want to, get there by the end of the semester, okay? So this is endoplasmic reticulum. It's just, reticulum means like a bunch of channels and network, and it's inside the cell. Okay. Once again, the nucleolus will be producing ribosomes, and the ribosomes are going to come out from the pores. And take a look for a second. Some ribosomes will just be floating around on its own in the cytoplasm, but some of them will be attached to the endoplasmic reticulum, like that. There we go. So let's label them first, and then I'll tell you more. So these are free floating ribosome. And then we have ribosome attached to ER. ER is endoplasmic reticulum. When you look at this under the microscope, because of the presence of the ribosome, it looks very bumpy. And it looks very rough in texture. So we call them rough endoplasmic reticulum. Okay, RER. Please write the full name on the quiz when I ask you to label it. Okay, rough endoplasmic reticulum. So what do the ribosomes do? Well, whether they are by themselves or whether they're attached to the ER, their job is to produces proteins. Produces proteins. So how is it different at the rough ER? How is it different at the rough endoplasmic reticulum? So it works with ribosome to produce and package proteins into vesicles. I'll explain. Just copy it down first.
What are vesicles?
Yeah, I'll explain. Yeah. No. Far from it. We're 50%.
What?
I'm sorry. Okay, start on the next page and then reorganize it when you get home. I'll post this, of course, right? Sorry. We good, guys? With me? Yeah, I hope. Okay, this next part is hard to explain. Don't draw, just let me talk first, and then I'll give you time to draw, okay? I tried this yesterday. It was even harder, virtually. Let's see if I do any better in person. All right, so here I'm gonna zoom in. We're zooming in so many times. I'm gonna zoom in one more time here. And I'm gonna draw this thing, okay? Again, don't draw this, just watch, let me explain, and then you decide what to draw later. Okay, so this is like the inside of the rough ER. And then we're gonna have the ribosome right here. Remember, the ribosome, even though they look like really small dots, really they are like a burger. They really are. Okay, there you go. And there is going to be a ribbon of mRNA, right? If you recall, the mRNA is a copy of what? Exactly, copy of the DNA, right? Contains the instruction to make the protein. So the ribosome will read the instruction and then it will build the protein according to those instructions. Okay, so the instruction will tell the ribosome to put, this amino acid with that one, and you link them together, and then you have your protein. Okay, seriously, don't draw this yet, okay? Because I have to erase things after. I will let you know when I'm done. Okay, so this is protein. Now, once you finish making the protein, we know the protein is going to fold up, right? With the secondary structure, the helix and so on and so forth. And ultimately, it is going to become a three-dimensional shape protein. So this is fold up protein. We good so far? All right, now this next part is the confusing part. So what's going to happen is the protein is going to move towards the edge, and then it is going to push on the edge of the endoplasmic reticulum membrane. It's going to push on it. Okay, it's going to push on it like that, and then it's going to move in there. And then eventually, it will push so far out that the whole thing is just gonna butt off, and then you have the protein in there. The best way to visualize this is to think of blowing soap bubble, okay? Now you blow this way, and then the bubble comes, you blow harder, right? And then it goes, it goes like that, and then you blow even harder, then it finally detaches, right? Can you visualize that? That's what happens. Okay, except no one is blowing the thing, okay? It's just the protein is coming and then it just moves towards the edge. It kind of elongates a little bit and then it bugs up. You got it? Okay, so that's the function of the RAP-ER working with the ribosome. It's able to package these proteins into a bubble, okay? But bubble doesn't sound nice. So they have to come up with more complicated words, which is vesicle.
So
all right?
vesicles are bubble.
Yeah, they are. But please don't say bubble on the text, okay? I will be forced to take marks off. What about these guys? Well, they can make the proteins, but they are not in a bubble. They're not in a vesicle, okay? They're just proteins that will be floating around. Some proteins needs to be packaged, others not so much, right? Okay, that's just the way it is. All right, so copy whatever you need. Yeah, please. Okay.
There's more,
Oh yeah, there's more. Yeah.
but this is like silent.
Yeah, this is like a silent. Okay, now if you're planning for space, we're gonna draw one more thing here, and then we're gonna draw one more thing here, and then like a little bit more, okay? It's okay, you can draw this again when you go home. In fact, it's a good idea to reorganize the notes when you go home, so that you have more consolidation. There we go. Okay, let me label this guy for you. This is a vesicle. This actually does happen, all right? You can see this happening under the microscope. You don't have to take picture. I post it online, okay? Yeah. Mine will be high definition, okay? Yours will have like other people's head, me on the side. No good. Are we good? Can I move on?
No.
No? Okay. Sure.
So,
Yeah.
if we don't use that again, we will have the NR and it's still not
Exactly.
They're just.
They would just be on their own. It wouldn't be wrapped nicely.
What would they say of function?
Yeah, those proteins will be able to function on their own, right? This one needs to be wrapped because it actually needs to be processed further, and that's what I'm going to show. It says vesicles with proteins that buds off BUDS off from the rough ER, okay? Here. Vesicle with proteins that buds off. I swear that's what they say in the textbook, okay? Buds off from the rough ER. Okay, that pinches off from the rough ER. Okay, I'm gonna move on. Now, you can guess, right? If there's a rough endoplasmic reticulum, there must also be a, what's the opposite of rough, huh? Smooth, that's right, smooth, okay? So, we're gonna use a different color, and generally on a picture, the smooth ER is further out from the rough ER. It's an extension of it, okay? And then you can just draw a few extra one here. There you go. All right, so these ones are smooth endoplasmic reticulum. Smooth ER. They do not have ribosomes on them. Okay, so without the ribosomes, without the dots, they appear to be smooth under the microscope. And that's why they're named the way they are. Smooth ER.
When we're going to make a break, I have
In 5 minutes-ish, yeah.
Yeah, it's not concerning details, something else.
Sure, yeah, of course. So, rough ER makes proteins with the ribosome. Smooth ER, what they do is they produce and break down lipids. Okay, lipids. Give me some example of lipids. Just yell them out. Triglyceride, good. What else? yeah, phospholipids, steroids. Right? So all these things, they are made in the smooth ER. Smooth ER. They also help you detoxify drugs. Okay, so if you are taking drug, I don't mean like, illegal stuff, like, regular medications, right? That also needs to be metabolized and detoxified. That's going to happen with the smooth ER. Smooth ER. So one of the things I want you to be mindful of as you go through these things is every cell in our body would have the same set of organelles, okay? They will all have the nucleus, they all have rough, smooth ER. However, depending on the function of the cell, they might have more of one type than another, okay? So what organ in the body detoxify drugs for you? Do you know? The liver, that's right. So liver cell, you would expect to have more smooth ER. Okay?
No, just drugs and alcohol.
That's right, and alcohol, exactly. Yeah. Okay. Are we fine here? Can we do one more thing? Yeah, that wasn't a question. It was just like rhetorical statement. We're gonna do one more thing and then we're gonna take the break, okay? And we're gonna use green right here. There's gonna be another stack of membranes that's not attached to any of these stuff. They are kind of on their own. And we call these things the Golgi body. This is the last thing before the break, but we have more afterwards. Oh, no. Yeah, we have more things. Okay. Yeah. Yeah, on the right side. Yeah. Sorry. I'll show you. Okay. I'll show you. Okay, there. So we are here right now. We just gotta write all these things and then we're done. Okay. This is something separate. You can start a new note. Okay. Yeah. Okay, we're almost there. Okay. Hey, you guys only have to draw it once. I gotta draw it like, 6, 7, 8 times every year. They are not supposed to touch, that's right. Yeah, they are like pancakes, okay? One on top of the other, except they're not touching. Okay, this is Golgi body, which is also known as Golgi apparatus. In older textbooks, they might say Golgi apparatus. And apparatus is basically like, multiple moving parts, right, so to speak. All right. The Golgi body is like the post office, and they have a slogan. They sort, modify, and repackage. Proteins. So you remember the vesicle that was created from the rough ER, right? It's gonna go to the Golgi, and it will be received by the first membrane, and then it will get passed forward, and sometimes it might go backwards a few times. So back and forth, and the content will be sorted, right? Like a post office, I keep on saying, right? You gotta sort the letters, right? And then you gotta modify them, and sometimes you need to repackage it, right? So like if the post office receive a package and the wrappings are falling apart or whatever, right? It's not safe for transport, they will repackage it for you, right? They actually do. So that's what happens. And in the end, after, moving back and forth a few times, then you are gonna get a brand new vesicle, okay? And the protein inside will now be modified, okay? I'm gonna choose a different color for that. Doesn't really matter. Okay, there we go. So this is modified. And there are a few destinations for this guy, and we'll talk about it after the break, okay? So you know the drill, not too long. We'll come back at 1:35, okay? 1:35. I'll leave this up on the screen. Okay, let's take a look at the PowerPoint slides for the stuff we talked about. It's pretty much the same thing with a few extra details added. So, RAGI right here, you can see is an extension of the nuclear envelope with the ribosomes on them, okay, the yellow dots. And further out, you can see that's where the smooth ER is, right, without the ribosomes. And this is the, once again, professional drawing. You can see the RAP-ER is going to work with the ribosome. Ribosome will be making the protein directly into the ER. And then the protein will move to the edge and the vesicle will butt off from the edge. And where is this going to go next? That's right. It's going to go to the green thing that we drew, right? The Golgi body, right? For sorting, modification, and repackaging, okay? So that's the vesicle. You can see there are multiple vesicles coming off. They will all be destined to the Golgi. The smooth ER has no ribosomes on them. Again, it's further out. That's what they actually look like, you know, with high power microscopes. The smooth ER main function is the production, the synthesis of lipids, including the things you guys mentioned, right? And steroid hormones. So you would expect the cells in the ovaries and the cells of the testis to have more smooth ER, because that's where some of these steroid hormones are produced, right? You remember sex hormones are steroid hormones, right? The testosterone, the progesterone, they are made in the ovarian and testicular cells. So it's not surprising you would have more smooth ER. In the notes, we wrote down smooth ER are also responsible for breaking down fat and detoxifying drugs. In fact, when a liver cell is exposed to medication in the bloodstream, for example, it will increase the amount of smooth ER it has to cope with that. Okay, so the organelles are not static; they could change when the demand varies. We drew the Golgi body, which are basically a series of membranes very close to each other. And you can read all these things, but basically is what I told you. The vesicle from the ER is going to come, and then it get passed forward and backward a few times, and then you will repackage them into a new vesicle. The next question that we're trying to answer would be, where will the proteins go from the Golgi body? Almost there with this note, okay? So we are trying to draw a cell, and I'm just gonna add the cell membrane here. And you know, you don't really have to if you don't have space, but like I'm just gonna put an edge around this guy right here. Okay, leave a little bit of space here. And there you go. Right? So this is the cell membrane, also known as the plasma membrane. And we are basically going to see where this thing can go. Right. So maybe you need the protein out of the cell. You need it in the bloodstream, for example. Then you're gonna send it out. So this protein will move to the cell membrane, and then it will get released from the cell membrane. So the protein comes out. Okay, the protein is gonna come out. All right, so this is destination number 1. Okay, out of the cell. Out of the cell. There, yeah.
This happens between the wrapped up protein and before the crumb stuff. Like, is that where you see this line from? Where that?
Oh, it doesn't matter. The chromosome stuff is just like a side note. Right. Just draw a membrane around these cells.
Oh.
Yeah, yeah. So like, just make sure you have an edge. You don't even have to draw the whole thing. Just make sure there's a cell membrane. The whole point of a cell membrane is to show you they can be moving out of the cell. Do you understand?
I
Yeah,
understand.
yeah.
Thank you.
Okay. Everyone else okay? All right. You need the stuff out of the cell, then you send them out of the cell, okay? Now sometimes, and I
Ask how much room do we need outside?
no, not much yet. That's it. Yeah.
No.
No,
Okay.
yeah. No, it could be the edge of your paper, okay? I know, yeah. Yeah, I know. Okay, so it can also become part of the cell membrane. Okay, part of the cell membrane like this. Okay, part of the cell membrane. So it turns out the cell membrane is not just phospholipids. We'll learn more about it probably on Thursday. It's made up of mostly phospholipids, some cholesterol, but there will be proteins in it as well. And the proteins are actually very important as part of your cell membrane. So that's destination number 2. Number 3, where else can it go? Well, it doesn't have to go anywhere. It can actually just stay in the cell membrane. Stay in the cytoplasm. Okay, stay in cytoplasm until it is needed. Sometimes you want to make things ahead of time. Right? You might not need it right away, but if you have the resources to do it, let's make some, and if we need it, then we will have it. Okay, so that's very common. For example, insulin is a protein, is a hormone that you need after you eat a meal, right? To help you absorb the sugar from the blood. You don't want to make the insulin after you have eaten. Okay? You want it to be already there, and when there's food in your system, then you can release it right away. Okay, so that's number 3. Oops, sorry. And then number 4, destination number 4, this vesicle will becomes an other organelle called, I'm running out of colors, guys. It's hard to believe. Whatever, it doesn't matter. Let's use hot pink here, okay? It's called lysosome. Lysosome.
It comes in what process?
It comes in what process?
Yeah.
Uh,
How it becomes.
Yeah, we don't have to know that, but there is, basically, remember the Golgi, right? The Golgi will sort and modify it, so it will modify it in a way that it will become the lysosome.
Okay,
Yeah.
so by the Golgi body.
Exactly, pretty much, yeah. So what does lysosome do? I'll just write down for you first, and then we're gonna go to the PowerPoints, okay? They are the recycling depot and the garbage disposal facility, basically. They break down macromolecules, right? Carbohydrates, lipids, proteins, you name it. They can also break down ingested material by the cell and old organelles. If something is not working and it needs to be broken down, it's going to go to the lysosome. Can also trigger something called apoptosis. I'll explain. Just write down first. And that's it. That's the end of the drawing, okay? No more. Until next class, okay?
This is something.
What is apoptosis?
Yeah, I'll explain, yeah. I mean, I think this is better than going through the slides alone, right? Like at least you will be able to, you know, hands-on draw a little bit, keeps you awake. Yeah, but not just macromolecules, it's also able to break down ingested material, okay, and all organelles. Can also trigger apoptosis. Okay, if you miss something, don't worry, it'll be online. So, all right, there you go. Yeah, there you go. Any quiz next week besides mine? Just wondering.
Yeah.
Everything? Nice. Yeah. Well, welcome to pre-health. Yeah. Okay, so lysosome. Lysosome has a lot of digestive enzymes. Let me remind you, enzymes are proteins that help speed up chemical reactions. So when you try to break down macromolecules, right, when you try to break down macromolecules, each reaction, each hydrolysis reaction will need to have an enzyme for it. If you want to break down the proteins into amino acids, you need an enzyme for it. If you need to break down starch into glucose, you need an enzyme for it. So all these enzymes will be inside the lysosome. Okay? So whenever there are molecules that needs to be broken down, it gets sent to the lysosome, and then the reaction would happen in there. Now, not all cells will do this. For example, in the human body, our cells do not need to eat food to get energy. Okay, but like if you're thinking about a cell that's living on its own, like the amoeba I showed you, then they will have to eat food from the surrounding, right? So when the food goes in and you need to break down the food, that's when the lysosome comes in. So when I say it breaks down ingested materials by the cell, that's what I'm talking about. There are some cells in our body that will eat other cells. We call them white blood cells. It's part of a defense system. So if it eats a bacterium, then the bacterium will be broken down by the lysosome. If you have damaged organelles that's too old or not functioning properly, then the lysosome will come and break it apart. And whatever resources was locked up by that organelle will now be available for other uses. So like I said, it's the recycling depot, right, of the cell. Now it's also capable of triggering apoptosis. That's a word that we will use again and again. Apoptosis is programmed cell death. Okay. Sometimes it is necessary to deliberately kill off cells, either as part of development or for the greater benefit of the whole organism. This is a picture taken from an embryo growing for 51 days. We were all once at this stage, and I don't know if you can see it too well. Let me see if I can dim the light a little bit. Yeah, maybe a little bit better. Okay, so you can see in this picture, these are fingers, right, by the way. And between the fingers, you see there are some web here. Yeah, so that's what happens. At some point during embryonic development, we have web between our fingers. But of course, when we are born, those webs are gone, right? Our fingers are detached, right? Which means the cells here needs to die off deliberately as part of normal development. Right? And so the cells would have to undergo apoptosis. Yeah. And the organelle that's responsible for that would be lysosome. Okay? Long story short, when you need to kill off the cell, the lysosome would release all of its enzymes, and then the enzyme basically would destroy it. Later on in the course, we will learn some cells are they contain too many mistakes, too many errors, and we cannot allow the cells to make more copies of itself, and then you need to kill it off. If we don't kill them off and you allow the air to propagate, you might end up getting more and more air, and that could lead to like a cancerous situation. Okay, so programmed cell death, where you deliberately kill off a cell, is called apoptosis, and lysosome sometimes has a role in that. Okay. Now, there are so many enzymes inside the lysosome, and since enzymes are proteins, which means they are coded by DNA. So if the DNA is mutated, it is possible for an individual to be missing one or more of these enzymes in the lysosome. When that happens, it leads to a bunch of conditions collectively known as lysosomal storage disease. Depending on which enzyme is missing, the outcome and the severity is going to be different. For example, in Pompe disease, if the enzyme that's needed to break down glycogen is missing, then the glycogen will not be broken down and it starts to build up in muscle cells. Okay? So it's not too big of a deal if it builds up in like your arm muscle and things like that. It causes them to become a little bit weaker, but it'll be okay. But if they start building up in your breathing muscle and your heart muscles, right, then your heart becomes very, very weak and your breathing is not enough to keep up, then a person could die from something like that, right? Similarly, in Tay-Sachs disease, which is one of the genetic conditions that can be screened early in conception, the lipid digestive enzyme is missing, and these lipids starts to build up in the brain cells, interfering with brain development. Okay. So there are many enzymes in the lysosome. If we're missing one or more of them, typically it will lead to diseases that are very detrimental, if not fatal, in early childhood. Okay? Yeah.
So do you have each lysosome for each enzyme or?
Oh, no, yeah, good question. The lysosome has all the enzymes all mixed in the same thing. Yeah. Okay, I will not test you on this, so you can cross it out. This was just for extra information, okay? No need to study. All right, this one. Let's see if you remember. You're gonna have to show me both hands for multiple answers. Yeah, yeah. I agree with one of them. Yeah, that's with the back. Really? No, no. That's correct. That's not correct. Anyone else?
Oh,
That's one of them. Yes. Yeah. One of them's correct. No. Yeah, yeah, that's right. This is not working out, guys. It's protein, right? You guys all agree it's protein? Yeah. What's the other one? RNA. RNA. Okay, you're not gonna find DNA except in the nucleus. Okay? You're not gonna find DNA except in the nucleus. Okay, how about this one? Yeah, that's one of them. Good, good, good. And both of you give me one correct answer. There are more than one though, yeah. No. Yeah, one of them is right. Yes.
this one, right?
Okay, okay. All right. So, Golgi is the post office. Post office do not detoxify for you, right? And it sorts, modify, and repackage. That's not the answer. RUP mixed protein, no detoxification. Lysosome, we just said it has a lot of enzymes in it, right? So it breaks things down. That's the correct answer. Mitochondria, we haven't even learned about that yet. That's not the answer. Smooth ER, also detoxification, right?
Yeah.
And so it's C and yeah.
If we only circle like.
On the quiz will always be one answer.
Oh,
Yeah,
okay.
yeah. On the quiz will always only be one correct answer. Okay, it's only when you do practice quizzes online and stuff like that, then you have more than one correct.
So what's the final answer?
Final answer is C and E. Okay,
Okay.
C and E.
So it has two answers here.
In this case, yes. Again, on the quiz and test, only one answer. All right. Yeah. Some of you were calling me over during the first quiz. It's like, I think I see an additional answer. Okay. If that's the case, most of the time is you got it wrong. Okay. All right. We're gonna do a few other organelles and hopefully we'll be able to wrap this up. Now we are done with the endomembrane system. Okay, we're done with the endomembrane system. It includes the nuclear envelope, smooth and rough ER, Golgi body, lysosome. Those are part of the endomembrane system. Everything we're going to talk about for the rest of the class is not part of the endomembrane system. And
So it means that it's not part of the next test.
no, it is part of the next test. Yeah,
Oh,
it is. Everything on lecture 3 is going to be on the next quiz. There are some exceptions, okay? I'll cross out a few more slides for you, right? So stay tuned. All right, this next guy is called the peroxisome. So it's really difficult to tell if I just show you a picture of a cell. Like how are you supposed to know this is peroxisome and this is lysosome if it's not labeled, right? Okay, they are both circle. Well, it's really hard to tell. So I'm not gonna ask you to label these ones on the test. And the functions are actually very similar, right? The peroxisome is going to help detoxify poisons, right? Such as alcohol, right? But what makes them different from one another is how they break down these toxins. For lysosome, it uses enzymes. We talked about that already. For peroxisome, which is this guy right here, it uses a special kind of reaction called oxidation reaction. We'll learn more about oxidation reaction in lecture 5. For now, oxidation means removing electrons. When you remove electrons from a molecule, you can break it down. And in this case, it releases a byproduct called H2O2, which is hydrogen peroxide. Hydrogen peroxide is toxic to cells, and the reactions must be contained. So these reactions are very damaging to the cell if you let it happen in an uncontrolled manner. And that's what the peroxisome is. Okay? You keep these dangerous reactions inside the peroxisome. You can still carry them. They are important for the cellular function, but you don't let it go everywhere in the cell. Okay. I guess you can think of it as like there are power plants that are like nuclear power plants. The reactions are dangerous if it gets out, but if you let it only happen at the power plant, then it's a controlled environment, right? And that's kind of what happens. It's a necessary process, but it's dangerous for the cell. So you have to keep it inside these peroxisomes. All right, this was in the notes that we wrote, the destination of those proteins. Okay? Which one is not the answer?
Not like.
Yes, it says except. Good. Anyone else? Yeah. Yeah, you guys are running out of steam. Huh? Yeah, good. This is the correct answer, right? This is the exception. Okay, so it can go into the cell membrane, it can become part of the lysosome, or stay in the cytoplasm, but it will not be the peroxisome. Remember, the peroxisome is not part of the endomembrane system. All right, we're just gonna go through a few more slides and then I will send you on your way. All right. Mitochondria is the next thing. All right, let's draw the mitochondria together because later on in the course we lecture 5 we have to learn a lot more about what's going on in the mitochondria. And so you need to understand how it looks like. All right, so this is mitochondria. Don't worry, this is a small drawing, okay? Small drawing. So singular is ION, plural is IA. English is difficult language. Not everything plural is just adding an S. Sometimes you have to change it completely. So here we go. Mitochondria. First of all, what does it do? Produces energy for the cell. So everything you do pretty much requires energy, right? To move the muscles, to breathe, for the heart to beat, to think, right? All those activity requires energy, and ultimately the energy comes from the mitochondria. Now the mitochondria is oval shape and it has two membranes. We have an outer membrane here, outer membrane. And then on the inside, there is a second membrane. But this membrane is going to be all folded up like so. Going to be all folded up. All right, so here, this is called the inner membrane. And it folds up into these finger-like projections called cristae. Okay. If you have a hard time visualizing it, it's like this, okay? So like this is the membrane, but then it folds up into these shapes. Okay, so it keeps on folding and folding and folding. So you can pack a lot of it inside the mitochondria, right? That was not a very good fold. Let's try to put them in the piece like this. So it folds up into this thing. Okay. And it just like wave-like all the way inside. So by forming these cristae, okay, what it does is it increases surface area for reactions. Okay. Energy production is basically chemical reactions. And if you are able to provide more surfaces for the chemical reactions to take place, then you're going to be able to produce more energy more efficiently. Okay. So that's why it's all folded. Can you visualize this? Two membranes, right? One on the outside and then another one on the inside. Now the gap here between the membranes, you have a gap. That's called the intermembrane space. Intermembrane space. We will learn about its function later on. Last but not least, on the very inside of this thing, it is called the matrix. The matrix. Yeah, like the movie. So cellular respiration is the reaction for producing energy. We wrote it down earlier in lecture 2, but I'm going to write it down for you again. It is glucose plus oxygen. You get glucose from your food. You get oxygen from the air you breathe. And it produces carbon dioxide. plus water and energy. Right? So this process, this cellular respiration, part of it takes place in the mitochondria. What kind of cells would you expect to have a lot of mitochondria? What's that? Sorry. Lungs? Yeah. Could be. Anyone else?
Muscles.
Muscles. Right? Muscles would be correct. Right? Every time you move your muscle, it needs a lot of energy, right? So heart cells, they have lots of mitochondria. Brain cells have a lot of mitochondria. And whereas something like a fat cell, they are mostly for storage of energy, right? They're not going to be producing too much. All right, I think that's a good place to stop, guys. Thank you. We will continue on Thursday. We will do lecture 4 as well, so print the notes. All right. See ya.